Good morning, everybody, and welcome to this seminar. So, as usual, it's going to be a webinar mode. So, for people online, hopefully you hear me. And no? Again? Do you hear me? Yeah, okay. So just report any issue on the chat if there are some online, and also you're not allowed to interact during the seminar, but you're allowed to ask questions at the end of the talk. So please put your question in the Q&A tab during the seminar, and we'll open your mic in the end. And also, I would take the opportunity to make a quick announcement for the TRALA conference that will be on the June 13th. And the speaker this year will be Göran Arnqvist from Uppsala University, who is working on sexual conflict and sexual selection. And now I will let Thomas present.
I think the computer is making.
Yeah.
Morning, everyone. So I'm really pleased to welcome Asher Cutter, who's coming from University of Toronto in Canada. And so since we've done the open policy right now for STEM, which is a good thing, it's great to have some international colleagues sometimes. And in fact, it's through the opportunity that he's going to another conference that we were able to distract him to come over just a little bit to talk to us about his research. And he gladly accepted, and thank you for that. And so to briefly introduce Asher, so he's been doing his PhD in Arizona, and he already at that time started to work on Caenorhabditis, on worms, and since then I think been following career on those creatures. And so after that PhD, he went to do a postdoc in Edinburgh. Work with Deborah Charles works, and apparently you brought your worms there in the botany. Well, Deborah Charles was doing botany most of the time, and to do experiments there and to interact with many people that were in the lab at the time, including many people that are here from that time when you did the postdoc in Edinburgh. And after that, in 2006, you were hired as a professor, associate professor, probably at the University of Toronto in the Department of Evolution and Ecology, and you've been there since then and working on those Caenorhabditis across many topics, mostly genome evolution, speciation, and also sperm evolution, and so I think, and there is apparently new stuff on microRNAs and many topics. And so that's kind of a mixture. I don't know what we're going to talk about, but probably a mixture of those. Thank you again for coming here.
All right. Well, thank you all for coming. Thank you for hosting me. It is a pleasure to be here. And I'm going to talk about a few different things that kind of connect across different areas in my lab. With the hopes that maybe some of them might be interesting to everyone, even if not all of them are interesting to any one of you. But they all are on this theme of looking at these Caenorhabditis nematodes as a system to think about diversification in different ways. And if you don't work with Caenorhabditis, you may know of C. elegans, the model organism. And if you've seen worms, you might think, well, if I've seen one, I've seen them all. So is there really any diversity here to study? And so I really like this quote from Eric Haig from a while back, where he says, if they all look alike, you aren't looking hard enough. And so I'm going to be looking a little bit harder today and some different axes to try and convince you that there's some really interesting biology in different ways. So one of the ways that we could imagine looking at diversity is in terms of DNA sequence polymorphism. So this is a plot showing DNA sequence variability, neutral polymorphism for a bunch of different groups of organisms organized by phyla. You'll notice the log scale. And the last column here are nematodes. In fact, actually, every one of those data points is a species of Caenorhabditis. So each one of these points is a species. And you'll notice that within this one genus, you have an enormous span of within species genetic diversity, ranging from among the lowest to, in fact, the highest levels of genetic variability of any known animal or plant. I'm not going to dwell too much on this except to kind of highlight that point. Another way you can think about diversity is in terms of species richness. And so discovery of Caenorhabditis species got off to a slow start about 100 or so years ago. But in the last 20 years has seen this really accelerated increase in species discovery. So we now have about 80 different species in culture that we can work with. And I'm showing here a recent phylogeny of a bunch of them. They're found throughout the globe, and in fact, including from some field work that I did with Chris Brendel at University of Nice in French Guiana at the CNRS field station there. You can find about 9 species in that one nature reserve. And I don't know, maybe half of them were new to science at the time I discovered them. So I'll come back to some of the work that derives from some of those samplings there. There's also interesting biology related to mating system. I'm also not going to dwell on that too much for the purpose of today's talk, but there's been 3 independent origins of self-fertilizing species, most of which have very broad geographic distributions, but the vast majority of species have separate male and female sexes. So for the plant people, they're dioecious. For the animal people, they're gonochoristic. So typical of what you might think of many other mammals. And I want to walk through very quickly one way that we kind of go about actually discovering these species, because morphologically, many of them are actually indistinguishable. The classic taxonomic characters, you can't really tell apart many of the species. And so this is a story of a former PhD student where we had some samples. They all kind of looked alike. You could get cross progeny between different individuals. But when we did a phylogeographic study looking at some DNA sequence variation, they clearly clustered into 2 groups. So for the kind of molecular evolutionists here, you have a KS of about 20% between these 2 groups. So quite high DNA sequence difference between them, which made us wonder if in fact they were different species. So we did some crosses, and then lo and behold, the F1s had much reduced fitness and really strong fitness reduction in the F2. So this seemed, using kind of a biological species concept definition, that these were clearly distinct species. And we've gone on to do a bunch of other work to kind of document some of the characteristics of that. So for example, you have this asymmetric pattern where when you have one of the parental species as the maternal parent, you have much more severe effects in hybrids. And you see that in reduced production of hybrid males. Those males they do produce tend to be, are almost always sterile. When you look at embryonic arrest in the F2 generation, it tends to be earlier when you have this particular cross direction. And when you look at what might be causing the sterility, it seems to be a combination of multiple things, including defects in the morphogenesis of the gonad, problems with the development of the sperm cells, as well as in male tails, that's their reproductive structure, and there are some abnormalities there. And so you can kind of look at that microscopically. And I'm putting these up as kind of a prelude to what I'll get to in a moment. So this is mostly published work, and so I just want to highlight some of the kinds of things we can get at. We can really look inside the embryos because the cellular development of Caenorhabditis is so well characterized, and look at what stages of embryogenesis arrest when you have problems in crosses between species. So I've kind of summarized some of these differences. It turns out one of the things that is distinct from these problems in hybrids is that actually there's no significant barrier to mating between the species. If you kind of put males of one species, females of the other together, they'll mate readily. If you put males and females of both species together on the same environment, they will also mate readily. So there doesn't seem to be any mating barrier. But this is just one case study. And so what we've been trying to get interested in is how general are these kinds of patterns when we look at a single pair of species. So
And in particular, what's the time scale of evolution that leads to these kind of reproductively isolated groups? And so one approach that we've been taking to do that is takes inspiration from some of the pioneering work by Coyne and Orr, where they worked in Drosophila and plotted some measure of how reproductively isolated different pairs of fly species were as a function of the genetic distance between them. So as a proxy for the amount of accumulated genomic change or the amount of time that had elapsed. And subsequently, people in flies have kind of gone on to look at this in more sophisticated ways. For example, separating out problems that arise in hybrids by sex, so males and females. How does that accumulate as a function of time? And when you look at different types of reproductive isolating traits, so if you look at sterility versus the inviability of hybrid individuals, do they accumulate at different rates in differences between sexes? So we're curious to see if there were similar kinds of patterns in worms. And so the first thing we needed to do was kind of think about this x-axis. So evolutionary distance. We could use just absolute genomic difference, but one thing we were keen to try and figure out is if we could actually estimate divergence times in these groups. And this is a really tricky thing. There's no fossil record for worms, but one advantage we do have are experimental evolution studies using mutation accumulation to document and quantify the input of new mutations on a per generation basis. And so we want to kind of exploit that as a way of connecting mutational change to the accumulation of substitutions. So I'm going to walk through how we go about doing that. There's another kind of complication in that, I don't know if you can make out this x-axis here, but I have shown here time measured in units of generations, and I'll show you how we kind of generated these estimates of divergence time. But we're talking over 1 billion generations ago as our estimate of the root of the group. So that's a very long time ago in units of generations. You do kind of a guesstimation that maybe they go through 10 or 20 generations a year, then you're talking about a common ancestor within this one group of about maybe 150 million years ago. So relative to mammals, that's a very long period of time for a group of organisms that are mostly morphologically indistinguishable from one another. Okay, so how did we go about doing this? So one problem is that if you compare DNA sequences to one another, ideally, if we were going to apply kind of a molecular clock type of approach, we would want neutrally evolving changes to accumulate. But when you compare the genomes of most species, actually they're saturated with substitutions. And so we can't really use that comparison for most species. So we have about a dozen pairs that don't have this saturation with mutations. So we can use them as a starting point. Another interesting kind of molecular evolutionary feature is that actually, if we use this kind of measure of divergence at kind of neutrally evolving sites within DNA sequences, within genes in particular, genes that have higher expression tend to have stronger codon usage bias. So this is kind of over on the left side of this graph here. So there's basically, this is evidence that there's selection that slows down the accumulation of some substitutions, even in those wobble positions within coding genes. And so we wanted to account for that. And so essentially what we do is we make a correction to account for this weak selection on synonymous sites within genes. And so then we can get kind of a distribution of these measures of DNA sequence divergence across these different groups. And we can kind of plug it into a Bayesian analysis called BEAST, built on a species tree of about 3,000 orthologous gene copies that are common to the 50 or so species that we have in our analysis. And then we can connect this to time, just assuming a simple molecular clock where we've got these corrections for these kind of nuances of the molecular evolution based on this direct estimate of the mutation rate from mutation accumulation experiments. And so this allows us to get kind of an estimate of divergence times for different species pairs, and then we can extrapolate that up to all the species in the genus by looking at the amino acid sequence change, essentially calibrated based on these 11 pairs where we have synonymous site-based neutral molecular evolution. So, and this is robust to kind of using different subsets of genes and things. So we can help account for that. So that's kind of the first way that we've gone about trying to get, okay, how evolutionary diverged are different pairs of species. The next factor that we want to think about is this vertical axis here. So some measure of reproductive isolation between species. And it turns out these worms are kind of a convenient system to do that in because in practice, people use the biological species concept essentially to define new species. So we do a mating test in the lab of some undescribed species to figure out whether it's actually biologically distinct. And so that becomes part of the information in the literature. It tends to be relatively qualitative. In particular, you take known species A, unknown species B, intercross them and simply ask, can they produce fertile and hybrid offspring? And sometimes it's a little bit more information than that, but it's still fairly coarse. Nonetheless, we can harvest information from about 258 pairs of species that were looked at in this way. And we can have a measure of reproductive isolation in terms of problems arising in zygotes. So either you have embryonic arrest or larval death. And we can relate that to how much our estimate of how much time has elapsed between the pairs of species being looked at. So essentially we have conspecific control crosses where there's low, if any, problems in producing next generation of offspring. But most species pairs are completely reproductively isolated from one another. And so we can fit a logistic function to this to see that within this kind of narrow, relatively speaking, time frame is when you get the accumulation of these barriers for embryos to be able to develop. We can also look at other kinds of things. So for example, will different species mate with each other if you give them no choice and you put males of one species and females of the other together in the same environment? And at least using that coarse kind of measure, you can see that actually they will quite readily mate with one another for very long spans of evolutionary distance. And so this is quite different than the story that you often see in many other organisms. So where actually you can get mating barriers to reproduction that can evolve quite early on. So this is kind of a contrast to that kind of system. So a couple take homes we can get from this is first that at least when measured in this way, this kind of post-zygotic reproductive isolation where you get developmental defects in hybrid embryos seems to evolve faster than these kind of pre-mating kind of barriers. A limitation is that these are fairly coarse indexes of reproductive isolation, and we don't have phylogenetic independence in these data points. They're pairs of species. Some of the species are represented multiple times on each of these plots. So that's something that we've been in recent time trying to improve upon. And so a current PhD student in my lab, Maya, has been looking at this in a more quantitative way. So rather than just a simple index, making a measurement, for example, of things like what fraction of embryos actually arrest when you make an interspecies hybrid for select phylogenetic pairs of species. So they're phylogenetic independent contrasts in a sense. And so these are some of the data where she's focusing really on that more recent time scale over which this accumulates. And there's a couple things you can see. One is that often the two cross directions differ from one another. So you have one that has more extreme embryonic arrest than the other, and also that it can be fairly variable. Another way that we're trying to dig into this is to take advantage of some of the developmental knowledge of this system and the ease at which we can look inside the embryos and ask, okay, of these embryos, that fail to develop at some point, what developmental stage is that at? And so here we're looking at, of those embryos that arrest, is it at the 2-cell stage or the 4-cell stage, or between 50 and 150 cells? It's set phases of development. And so we're still in the process of collecting more of these data, but so far, what we find is that most of them have a peak of embryonic arrest during gastrulation. So it's not that the species that share a common ancestor more recently have arrest later in embryonic development than those that arrested farther apart from one another. So there seems to be a hotspot during embryonic development that is sensitive to perturbation by putting the genomes of two different species together in the same cells. So I don't have any data yet to show you, but Maya is also collecting information on when we mix males and females of two species together in the same environment, can they detect one another as different species and mate assortatively? And because worms, they're all kind of transparent and look similar, we take advantage of a trick of staining males with a fluorescent dye that also stains their sperm. And we can see that sperm get transferred into other individuals. And by distinguishing the sexes with fluorescent stains, it lets us tell how often they mate with the same species versus a different species when they're exposed to both possibilities. And where eventually I want to kind of take this is to try and connect up the idea of how reproductive isolation barriers relate to the developmental process, and whether there's any predictability in the kinds of genes or the kinds of phases of ontogeny that are most susceptible to this. And we might be able to get some predictions about that. So for example, if we look at genes that have different profiles of expression over development, so the x-axis here is basically going from early embryogenesis to the adult stage on the far right. And you have different types of genes that have kind of different stereotypical patterns of expression that may peak in late embryogenesis, or peak in early embryogenesis, or peak in the adult stage. And if you look at the rate of protein evolution, you find that actually a subset of these evolve much faster. Those that have their peak expression early in embryogenesis or in the adult stage have much more rapid protein sequence evolution. And to the extent that different proteins interact with DNA to control expression, they might be predisposed towards creating genetic incompatibilities in hybrids. So that's like one way we could think about it. Another way is to think about gene expression in hybrids. So this is from a study from a couple of years ago where we, in this case, used whole worms, and we measured expression in each of the two parental pure species, as well as in their F1 hybrids. Using RNA-seq, we can distinguish the expression of the alternative alleles in the hybrids using allele-specific expression, and look at how, and using some inferences based on approaches developed by Patricia Whitcock and other folks, infer whether or not there have been kind of cis-regulatory changes or transacting changes or some combination of both. So again, if you have divergence in the molecular controls of gene expression, that's another way that in hybrids you could get disruption of expression in a way that causes developmental problems. Okay. So I've talked a bit about how we can get kind of reproductive barriers between species and sort of how development works. One phase that I haven't really focused on, though, and sometimes it's quite tricky, is what happens not if there's a problem with mating per se, but there might be a problem with fertilization, okay? And so this is kind of a often cryptic period in the interaction between different species. And yet oftentimes the cell types and structures that are involved in those interactions after mating can evolve quite quickly. And so they're especially interesting in this context. So it turns out that if you look in any particular spot in nature, oftentimes you can find multiple different species At least for some of them, we've shown that they are somewhat indiscriminate in which species they'll mate with. And so what we wonder is what happens when the gametes from one species encounter a different species. And about 10 years ago, we first looked at this where we were focusing our attention on this one particular pair of species. It's called C. briggsii. It actually has hermaphrodites. If you leave it alone to self-fertilize, it'll make a bunch of babies. If you allow it to mate with a male of its own species, they get an additional transfer of sperm. And so they're no longer sperm limited and they make even more offspring. But if you take those same animals and mate them to a different species, they don't even use their own sperm to fertilize. They're essentially sterile upon mating with males of another species. So they don't even use their own sperm to fertilize their eggs. And if you mate them with males of their own species, this isn't really rescued by that effect. So there's some problem that arises in being able to fertilize by having mated with males of the wrong species. And in fact, when we look within their bodies, what we find is that although you see sperm in the usual locations in the uterus and spermatheca, the sperm storage organ, very quickly over a matter of hours, we observe sperm cells in ectopic locations. And I'll show you what I mean by that in a minute. And we think it's actually the sperm cells themselves that are what's driving this. And with my collaborator, Eric Haig, we did some RNAi manipulation to basically transform males into making only oocytes instead of sperm cells. And if you mate those individuals in the same kind of a setup, they have no effect on the reproductive output of those hermaphrodites. So it's something about the transfer of the sperm cells. They can also make seminal fluid, I should say. So we went on to show that actually this kind of sterilizing effect, actually we can observe in many pairs of species of Caenorhabditis. And it looks like this. So this is a time-lapse movie. Here's a worm, here's its head. The sperm should be these glowing areas in the uterus and spermatheca, but when you mate it with a male of another species, you may be able to see these white dots, which are individual sperm cells. They've migrated up into distal portions of the gonads. Some have even escaped the reproductive tract altogether and are crawling around the head region of the worm in the coelomic cavity. So this is a really kind of dramatic effect in some cases. And has really profound effects on the fitness of those individuals. More recently, we've been taking advantage of two species that we can find sympatrically in the Nordic Nature Reserve in Fenstianum. And so I'm going to tell you a little bit about this recent work. So the first thing we wanted to know is, okay, they cohabit the same kind of forest environment. If we use this kind of staining trick, can we tell whether or not they actually recognize each other as distinct species and will not mate? So we did a setup using this kind of approach and to orient you on how we looked at the data. Here we have, we used 3 different ratios of each species. So in one treatment, one species or the other was rare at a frequency of 10%. Another intermediate frequency where there were 25% of the rare species, and then kind of an even mixture. And then we look at the fraction of females that receive sperm from the wrong species. So if they were perfectly able to distinguish one another, we'd see all our data points on this purple line at 0 at the bottom. If they couldn't tell each other apart at all, it would be in this kind of indiscriminate mating region here in gray. And these are kind of bounds based on some resampling of how we set up our experiments to give us confidence intervals. And what we actually observe is something in between, where there's kind of some degree of assortative mating between species, but it's imperfect. So they make a lot of errors in mating with the wrong species. And this has pretty striking effects. So here we're looking at where do we find those sperm cells in different parts of the body where if it's benign, it should be in the uterus or spermatheca. And that's exactly what we find with content-specific crosses the vast majority of the time. But when this one species called Caenorhabditis macrosperma is the paternal sperm donor, and Caenorhabditis, the other species, is the maternal individual, you find that very frequently you find these sperm cells in these ectopic locations. And this has this really detrimental effect. So if you look at the lifetime fecundity of the pure species crosses, or when Caenorhabditis is mated with C. macrosperma, you see a really profound reduction in offspring production. Moreover, it drives a really rapid mortality of those females as well. So this is a survival curve in days following mating, either conspecifically in blue, or in this other scenario here where you get this really radical different survival curve. So another thing that I want to point out about this situation is that the species where the females are kind of sensitive to this sperm cell invasion, they actually, if left to their own devices, would have much higher total fecundity than does the species that causes this sperm-induced harm, okay? So if we were naively imagining they were just competing over resources, we would expect that the species that has the higher reproductive output would win in resource competition because they can produce more offspring per unit time in a shared resource environment. And so the question is, which one does win if we mix them together, right? So we have this harmful effect that males of one species can have on the other, but that might counteract the benefit, the competitive benefit of the high fecundity of that species that it influences. And so we set up essentially a very simple two-species community and looked at it over the course of about 7 generations and asked which of the two species essentially takes over that two-species community to look, or do they coexist? And no matter what starting density or starting frequency we used, we always found that the species that has lower lifetime fecundity but induces this reproductive harm on the other species, it always increased in relative frequency across this time. And so we did a little bit of modeling using a Leslie matrix approach to show that this reproductive interference was sufficient to kind of explain the changes in relative abundance of the two species. So we find this interesting phenomenon where actually what we think of as the worst resource competitor is actually able to invade the environment and take over. And this is another way of saying that we conclude that this reproductive interference effect, at least in this system, outweighs resource competition. And so we have this kind of effect of these individual sperm cells actually ramifying up to kind of a community scale of how species interactions work. Okay, so what's so special about the sperm of the species? So based on some prior work with Chris Brendel, we know that these very large sperm cells get their start at the very early stages of meiosis, and that persists throughout the sperm development. But what we really wanted to do was look inside the sperm cells to see if there's anything unusual going on there. And I'll get to that in a second. So as that one species may be obvious, the species named Macrosperma, large sperm, that's why it was named. And when we did a phylogenetic comparative analysis of about 30 species, we found a huge range in the average size of these sperm cells. I should mention that nematode sperm cells are amoeboid. They're like a little amoeba. They're not flagellated individual cells like you may be used to thinking about. But there's been multiple species that have these very large sperm cells. And when we looked at this across the phylogeny using a kind of an Ornstein-Uhlenbeck process, we inferred that actually there's been independent origins of this gigantism several times on the phylogeny. And this is kind of interesting in other ways too, because we also measured egg sizes. And so what this radical change in sperm cell size does not have an equally huge range of egg sizes. So if you quantify what the volume of a sperm cell is relative to an egg cell, you have a 20-fold range in what the degree of anisogamy is between egg and sperm cells. So in the extreme, with these species with very large sperm, you may have a 20-to-1 ratio of egg to sperm volume, whereas in the species with the smallest sperm, you may have a 400-to-1 ratio of egg size to sperm cell size. So we're really curious to know, like, what on the inside of these cells makes them different. So we've started doing some electron microscopy to try and understand if there's some kind of peculiar allometric relationship between the organelles within the sperm cells and the overall size of them. And it turns out that sperm cells are very simple. They actually get rid of most of their cellular contents. So all they're left with essentially are mitochondria, this specialized organelle called a membranous organelle that fuses with the plasma membrane and dumps its contents into the seminal fluid. And then, of course, the nucleus. And so we can easily try and look through and quantify them. And so my student, Rebecca, one of the things she did was just using cross-sectional analysis, kind of look at the number and density of these different organelles. And what she found, I don't want to dwell on this too much, but essentially what she found is that the species with the larger sperm in this two species comparison has more mitochondria, more of these membranous organelles, and a greater total area taken up by the mitochondria, in part because the sizes of the individual mitochondria are larger. And so we don't totally know why that might be the case, but there's some evidence in some other organisms that larger mitochondria have more active energy production. And so perhaps these larger cells require more energy demands for their motility. So the next thing that we have kind of ongoing now is teaming up with Mei Zhen's lab, who's a neurobiologist, and she's kind of developed this really cool technique for having serial slices through worms to then build up with an electron micrograph reading of a three-dimensional volume. And so we're starting to do this looking at sperm cells instead of neuron connections, so that we can actually do some of these quantifications in a truly volumetric way to look at the relative abundance of the mitochondria and these other organelles within them. So you can even see here that in this individual, so the spermathecal valve, where they would kind of go up through to be able to fertilize this developing oocyte, all the pseudopods are kind of pointed in that direction, which kind of helps you see how they can clean up the sense the right way to get into the pole position for fertilizing the egg. So this is still very much in progress, but we're hoping to be able to compare species using this volumetric electron microscopy. Okay, so here's a little bit more speculative. We have multiple species that kind of evolve these giant sperm cells, but they actually make fewer of them. So that seems kind of peculiar, and we someday hopefully would like to get at some experimental tests of some ideas about what conditions might favor that kind of evolution. So one idea is actually it might relate to mating group size. But the logic being, if you have a very large mating group with more potential mates, then selection actually might favor better insemination abilities. So the transfer of many smaller gametes. But if you have a very small number of mates, where essentially every individual mates with every other individual, selection may put a premium on the fertilizing ability of every individual sperm that might favor the evolution of these larger sperm cells that are more individually more competitive and more able to fertilize eggs. So we don't know. There may be some experimental evolution that we can try to use to get at this kind of idea, but we haven't kind of gone down that path. Certainly in some species that also happen to have very large sperm cells, we know something about their life history that might be compatible with this kind of idea. So this one species, C. japonica, that's found in islands off of Japan, has this very specialized relationship with this bug where just tens of individuals on the back of the mother will crawl off, on the back of the mother insect will crawl off as she lays her eggs in a little nest with these fruits. The worms will then mate and reproduce among themselves, among those tens of individuals for some number of generations, and then crawl onto the backs of the next generation to proceed. So they have very small mating group sizes in that particular species. And there are some interesting other correlated traits. So the species that have independently evolved these sperm cell gigantism are different from species that don't have these large sperm cells in that they rarely deposit a copulatory plug that may serve as mate guarding in species that have smaller sperm cells. Okay, so far I've kind of gone through talking about thinking about speciation in the context of Caenorhabditis, how that relates in some ways to sperm cell traits and interactions between species. I'm going to shift gears a little bit to this last section about thinking about small RNA evolution. And in a second, I'll point out that actually there are some relevant connections to both of these things that I've talked about so far in terms of thinking about speciation and sperm cell trait divergence. So what are small RNAs? Well, there's something that's involved in gene regulation, and it turns out there are a number of very distinct small RNA pathways that regulate gene expression, and they work through this class of effector proteins called argonauts. And so here's kind of a survey across many different groups looking at how many of these argonaut proteins they have encoded in their genomes. So you can see that plants have a fairly large repertoire of these microRNA related argonaut proteins. Most animals have a smaller abundance of both microRNA and piRNA or PB interacting related argonaut proteins. But when we look at nematodes kind of on the bottom of this figure here, there's been a kind of a huge expansion in particular of this one kind of novel class called worm-associated argonauts that work through a slightly different mechanism called endogenous small interfering RNA. So they actually use pieces of coding sequence that's been transcribed to interact with these argonauts to affect expression of, in an autoregulatory way, of the gene that they came from. So these argonaut proteins, they're also kind of interesting, and in addition to having kind of expanded in kind of their functional repertoire in nematodes, many of them have differential expression across different tissues and different life stages. So here's some work from Julie Claycomb's lab from Uri Sarusi, where some of these argonauts are expressed only in spermatogenesis or constitutively in the germline. Others are primarily somatic in their expression, and so there may be opportunity for different ways that gene regulation works through these different small RNA pathways in different types of tissues. And so the connection to loosely to the kind of first parts of the talk are actually some work by Zhongying Zhao in Hong Kong, where he looked at interspecies hybrids of two species of Caenorhabditis and found that one of the things that got disrupted was regulation by small RNAs. And it was this endogenous small RNA pathway, one of those. And one of the things that disruption of this small RNA pathway led to was sterility due to defects in spermatogenesis. And so this is kind of my kind of weak link to thinking about this connection between different aspects of some of the research that we have going on in the lab. So one thing that my student Daniel has been working on is trying to understand how, if you zoom in a little bit from across the tree of life to just within the Caenorhabditis genus, how have these argonaut proteins as a family changed? And so fortunately, we're able to use the reference genomes produced by the Wellcome Sanger Center for about 50 species that we had at our disposal, and Daniel identified over 1,000 argonaut genes across those 50 species that nicely cluster into these well-known kind of groups based on their function that's been described in C. elegans. And there are a few things that I just want to point out about this. So one, This blue star here, there's this branch here where there's what looks like the origin of a new subfamily of these Argonaut proteins. Structurally, if you plug it into AlphaFold, they look fairly similar to kind of Argonauts that have microRNA related function, but they tend to have either a reduced or truncated region of this one domain of the protein, which is very unusual. There are some archaea that have a similar kind of phenomenon going on with respect to that particular protein domain. But right now we're kind of treating this as kind of a hypothesis to look at in a little bit more detail as to whether or not this cluster of about half a dozen seemingly novel Argonaut genes actually are functional within the cells of the organisms, whether they actually act in gene regulation involved in small RNAs or not. And so for that, we're collaborating with Julie Claycomb, who also is at the University of Toronto, who's a specialist in Argonaut biology. So when you look across species, there's also huge variation in how many Argonaut proteins they encode, even just within Caenorhabditis, okay? So I've kind of color-coded them. Some of them are basically represented once and exactly once in the genomes of pretty much every species you look at, whereas others are super variable in their relative numbers. So any given species ranges from having 9 Argonaut copies to 46. And these subfamilies that are associated with these endogenous small interfering RNAs, these kind of worm-specific types of Argonauts, are the ones that are most variable in copy number. And so, I mean, we don't have a full answer, but in terms of thinking about what might explain some of the variability in this gene copy number, well, one simple thing is that genome size explains some of the variation. And so you get a positive association between genome size and the number of Argonaut gene copies, as you might expect. And that's true if you use a phylogenetically corrected analysis. And another possibility, given that some of these small RNA families are involved in regulatory control over selfish genetic elements, transposable elements, we thought that maybe the number of Argonaut gene copies might relate to transposable element abundance. And for the most part, actually, we saw no association. There was just one subfamily of these worm-associated Argonauts that was negatively correlated with the number of transposons that you find in the genome. So, and it's still a little bit of a squishy correlation. So we kind of speculate that at least for this class of Argonauts, perhaps having more copies allows better control of transposable element activity. Another kind of interesting point about this is if you look at the number of copies of different members of this protein family, you can see that actually this one Argonaut protein called PERG1, which is essential for the pi-RNA pathway, the Pee-wee interacting portion of this regulatory pathway, that there's no evidence in 4 of the species that this gene is present. And we've done a bunch of different analyses. Of course, it's tricky to know if we might actually have missed it, if the assembly is poor, things like that. But as far as we can tell, it's not there. And in fact, when we look across a number of other different proteins that are also critical in this pi-RNA biogenesis pathway, those species that are missing this one Argonaut are often missing other members of this pathway, in particular this PARN1 gene. So we think that that's additional evidence that this entire regulatory pathway has been lost convergently and independently in several different species. Now, these pi-RNAs themselves, they're actually little genes. They're little genes that encode an RNA as the functional transcript. And they have a very stereotypical upstream motif that's thought to be associated with the promoter. And so if you just look for this motif in the genome of C. elegans, you find that you have these kind of clusters where it's overrepresented on chromosome 4, which is where the pi-RNA genes are. So we looked for this motif in all the different species and found that those that are missing this pi-RNA pyrene argonaut also have a very low representation of this motif that's associated with pyrene. So we think this is additional evidence that actually they don't functionally regulate their genomes using pyrenes altogether, based on this kind of motif analysis as well as the proteins that are encoded. So I think this is kind of a fascinating case where you have, loss of an entire regulatory set of machinery independently in different lineages of a group that's quite closely related together. There are other species of animal that have lost this pathway, but this shows that even on much more recent timescales, you can see that happening. So we're really curious to also kind of dive into this in collaboration with kind of our functional biologist collaborators to understand what the consequences are for those species that have lost that mechanism of gene regulation. One final point on this small RNA stuff is that there's this one protein called Caesar1 that has two isoforms in C. elegans. So the long isoform has this extra exon that has these RG repeats. And it's expressed specifically in the germline associated with spermatogenesis, as well as in some parts of the soma, whereas the shorter version of the isoform is just expressed constitutively throughout the germline. And when we look across these species, we see that actually there appears to have been an evolutionary gain of this second long isoform in the ancestor of this clade of this kind of what are called the elegans group of species. So if you look in their genomes, they all have this kind of upstream exon that we think would allow them to make these two different forms. And if you do a kind of PAML type molecular evolutionary analysis to look for accelerated rates of evolution, you see that it's exactly on that branch that you get accelerated evolution in the protein sequence of this shorter form. And so together we kind of have this thought that that there may have been kind of the evolution of a novel isoform in this ancestry that allowed kind of functional subfunctionalization in an adaptive way for how it uses this argonaut and it's mediating small RNAs. So we've so I've talked to you a little bit about thinking about these kind of regulatory dynamism involving these argonaut gene families, how they vary across different species. And given that one way that you can cause genetic incompatibilities to evolve is through changes in gene regulation, we think that this may be an interesting kind of way to think about some of these differences involving small RNA pathways. And especially given that some of the argonauts have expression that's restricted to spermatogenesis, and in particular this novel or newly arisen isoform of this Caesar1 argonaut gene, it suggests that actually when we see sperm-related dysfunction in hybrids, maybe it could be mediated disproportionately by certain types of regulatory pathways. So one kind of way that we're trying to get at this, and so this is some unpublished data where we have these two species that we can hybridize. And what we've done is we've separated out males and females of each species and dissected their gonads separate from the residual somatic tissue. So remember, worms have about 1,000 cells. So it's a, although it's a bulk kind of set of somatic tissue, it's a very small number of cells that is actually associated with. And then we've done the same thing for the reciprocal hybrid crosses, where we've dissected out the gonad and somatic tissue separately for males and females and looked at their transcriptomes to use this approach where we can look at allele-specific expression. And I'm showing you kind of a diagram here to illustrate one of the things that we're most interested to divine from this kind of approach. So if you had two parents that had the same level of expression of some gene, but when you look in the F1 hybrids, you see that the gene copy inherited from one parent had much higher expression than the other. You have this kind of bias in the allele-specific expression in the F1 individuals. And this can essentially reflect they're having, even if the average level of expression is the same in the hybrid individual in both parents. And one thing that this can reflect is having divergence in both like a cis-regulatory element that controls the expression of the gene, as well as some transacting factor that's encoded elsewhere in the genome that have evolved to compensate for one another. So even though the two species don't differ in this trait of expression level, it's kind of hiding this under the hood molecular evolution of the regulatory controls that evolved to compensate for one another. And so by looking at expression in the two parental species and in the allele-specific expression in hybrids, we can identify genes that have those properties. And when we do that, what we find is that actually those that have these kind of compensatory interactions are overrepresented among those genes that are expressed in males, in particular, for both gonad and soma. So I'm not showing you the data here, but actually what we find is that we generally see greater misexpression of genes that overall have biased expression in males, but the amount of misexpression is actually greater in females. And yet it's the females that show less morphological or outward dysfunction in their development. And so one conclusion we draw from that is that the gene regulatory networks involved in that involve male biased genes are kind of more sensitive to perturbation than are the gene regulatory networks that drive development of female individuals. So the females are able to actually tolerate more misexpression and still generate kind of a robust phenotype. So one direction we're kind of will be next step for some of this analysis is actually we sequenced the transcriptome to very high depth so we can actually try and detect alternative splice forms. And so to look at this kind of approach where you might get different splice forms expressed differentially by which parent they happen to come from. So I think there'll be a fun other direction of thinking about gene regulatory change. So just to sum up, I've kind of tried to walk through a few areas of kind of research in my lab, some of which is already out there in the literature, some of which is still ongoing. Thinking about speciation, where I told you about how we can think of the accumulation of reproductive isolation between species, how there are lots of different traits that can get disrupted, and how we showed that from our analyses so far, it seems like actually embryonic development or post-zygotic isolation seems to accumulate problems more quickly than barriers to mating with different species in the first place. I also talked a bit about kind of the sperm biology and this divergence in this one trait across the genus that otherwise is mostly indistinguishable morphologically from species to species, where we've had the independent evolution of sperm gigantism multiple times, and how this kind of ramps up to affect interactions between species with this reproductive interference that can out control even resource competition. And then actually I went into these small RNAs where this really dynamic evolution of this Argonaut gene family involving gains and losses and changes to the function, presumably the functional repertoire of the ways that gene regulation can work. So that's it for me. Thank you for your attention, and thanks to the folks in my lab and collaborators and some of the different aspects of this work. I'm happy to take questions.
The beginning of your talk, and then also later, you talk about crossing these species one another and having some hybrid sterility. And I was wondering if you looked if transposable elements would be involved in that.
So are transposable elements involved in the sterility of hybrids?
Yes.
Because we've not looked at that directly. My recollection in our transcriptome analysis of the F1s is I don't believe there was anything obviously different in terms of expression of the transposable elements, but So I think I don't think we're seeing a pattern like sort of hybrid dysgenesis that's been seen sometimes in between population crosses of some Drosophila.
And do you record some transposable element being different? The population being different?
Yes, so the types of transposable elements often differ in the genomes between different species. So one possibility is maybe the F1s are fine, but maybe in later, if you were able to get F2 or later generations, then there might be some disruption of transposable element activity, but we haven't looked at that.
Yeah. Sorry, is there anything known about the ecology of these species, whether they live in some sufficiently different niches so that they don't encounter each other often enough, which might perhaps explain why when you put them together, they mate?
Yeah, so that's a very good question. So I said that those that one species pair we work with are sympatric in the sense that we can find them in the same forest. But actually, where you typically find them are on rotting fruits or rotting flowers that are, you can think of as patchy resources and very ephemeral. So we think that there's probably a very strong kind of metapopulation kind of dynamism to their movement. And within any one patch, there may not be very many species, or may be unlikely to be colonized by multiple species. So it may be that there's at a very small scale, they're kind of allopatric and not encountering each other very often. So that's one possibility, but I have no direct evidence for that. Yeah. So I think it's very plausible that even though you can find many species in the same area, perhaps they don't have these pre-mating barriers because they don't encounter one another very regularly at that very tiny scale of tiny little patch. It's possible.
Perhaps just to follow up on that point, because it's very surprising that there is such a long time and an absence of pre-mating isolation. And so following up on that idea, you know, in studies on reinforcement, sometimes you see that pre-mating is stronger when species actually encounter. Would it be possible that here the reinforcement takes the form of this reproductive interference, and that, in fact, it's not just a byproduct of sperm cell not recognizing things, but actually a locally evolved interference? Would it be a possibility?
I mean, maybe I haven't modeled it in terms of, I mean, so one idea is you could almost imagine that this sperm-induced reproductive interference is like a weapon of competition between species, essentially.
Is it stronger than locally?
Yeah, I don't know. Yeah, I mean, I think I don't know the relative plausibility of that versus just kind of a within species sexual selection, sexual conflict kind of issue that leads as a byproduct this interspecies effect. I don't know what the relative likelihood of those is.
Did you investigate how reticulated is a phylogenetic tree, and if it can produce problems to the species identification? And actually, do you have to sequence the genome to identify a species, or do you use markers or something like that?
Yeah, so there's several things in there. So one, depending on the node, there can be a lot of gene tree, species tree discordance. And especially where there's a very short internal branch, there's very high discordance. It doesn't actually matter too much for the estimates of the divergence times. So we've tried using different topologies and seeing how sensitive it is to our inference of the divergence time. So it seems fairly robust in that way because it just kind of moves it to another branch in the tree, but the total distance from the tip is pretty much the same. The other part of your question in terms of like how do we distinguish species? So the practical way that we go about identifying species is we go in nature, we collect a sample, and we want to know if it's a species that's already known or something new. So one is there's a barcode sequence that we can get to see, okay, roughly where is it in the phylogeny? And then we just take those 3 or 4 species and do mating tests, so crosses with the unknown species to see if you can get fertile next generation offspring from it. So that's the practical approach to identifying a new species. We're not using genetic distance to determine species identity. Did I get all the points? I'm not sure.
Do you have surprise sometimes? Genetically, it seems to be the same species, but when you do crosses, it doesn't work.
I think most all of these species, the genetic distances are so deep that it's not that surprising. We have some very experiments from 2 weeks ago that where we thought essentially we're seeing some artifacts with our conspecific crosses that we thought were due to inbreeding depression in the strain that we're using. So we started doing some inter-strain crosses of what we thought were the same species and seeing that certain combinations of them had problems in hybrids. And so I suspect that when we look more carefully, there may be kind of incipient species or subgroups that are more genetically related than we realized. But certainly when we do kind of polymorphism analysis, there must be an incredible amount of ancestral polymorphism that's shared even between them.
Maybe you said it, but I missed the point. Can the giant sperm can transfer mitochondria?
I did not say, and I don't know the answer to that, but I'm very curious to know. I mean, the dogma is that everything in the sperm cell except the centrioles and DNA gets degraded in the early zygote. But especially in a species where it's actually quite a large volume relative to the zygote volume that is transferred paternally, we don't know if it's totally efficient at degrading mitochondria and other things that are transferred. There certainly are actually some paternal effects, small RNAs that get transmitted through sperm cells that may influence development in some way, at least early on in development. There are also some interesting toxin antidote selfish genetic elements that get transmitted randomly through the sperm and lead to basically you require a kind of transmission of the maternal antidote to counteract the toxin. So it's possible that not everything that gets degraded, and I have had on my to-do list to check about the mitochondria in particular, but we haven't actually done those experiments yet.
You have no question right now? Are there any other questions?
Maybe about these species that lost the entire regulatory pathway. Any speculations whether that is it perhaps related to the high number of these RNAs?
Yeah, I honestly don't know. So, I mean, one other kind of answer, actually, to your previous question that may be relevant is about the ecology of these species, where it's mostly unknown. Many of the species have been found once in one place. Some of them have exactly one isolate in the lab even, so not even kind of something that you can do a population genetic study on. So my first inclination would be to try and look to see if there were kind of parallel features of those species that might predispose them to kind of convergent loss, but it's I think it's tricky to kind of know how to do that. So my short answer is no, I don't know.
Just have a small question about almost the last slide or what the last results where you got these male reproductive genes in the where you got this cis-trans coevolution. Are those genes particularly located on the X chromosome?
So we've separated out the X, and I'd have to check. To do that kind of analysis on the X chromosome in males is tricky because it's homozygous in males. So you have to actually do your calculations in a slightly different way, and it's not a perfect apples to apples comparison to the autosomal encoded genes. I can't remember off the top of my head, but I can check. Maybe later today I can tell.
Anything else? Thank you again.